Morning all. We are back again to the last biomolecule, the nucleic acid. Very important biomolecule. Okay, all of them are important, but this determines who we are. So we are talking about the nucleic acid. If you talk about the nucleic acids, what comes to your minds are the DNA and the RNA. So we are going to see what do these nucleic acids do. They dictate, the first point, they dictate amino acid sequence in proteins. Still, when we learn about genetics, hereditary science, we talk about at the point where you inherit the characteristics or there is physical expression of a certain organism or a certain individual, or you can tell this is the son or the daughter of so and so because of the resemblance. We are talking about a phenotype, the gene product. That is the level of the protein we are talking much about. So it starts at the replication process where the DNA synthesizes itself. It moves to the transcription process where the RNA is synthesized. At the point the RNA is synthesized, there is always the messenger RNA which carries the codon. The codon are the three bases which represent an amino acid. So you'll have the messenger RNA carrying the codon, then you'll have the transfer RNA carrying the anticodon. So the three bases, which could be adenine, uracil if it's an RNA part, or guanine, the three bases will represent an amino acid. At the translation process, which takes place in the ribosomes, it will equate to an amino acid. Then remember we said amino acids are the building blocks of the protein. So you'll have a polymer of this amino acid, and at the end of it, it will be able to be dictated by the DNA and the RNA. So we generally talk about the central dogma theory. At the level when you are seeing the gene product, which is the protein, the phenotype can easily be seen. There is a genotype which underlies it. So behind every phenotype, there is a genotype which underlies it. There is a DNA, and only the DNA is hereditary material. So there is a genotype which underlies a phenotype, which you see. So behind every physical expression, there is a gene which is responsible for it. So they dictate amino acid sequence in protein at the point of translation, where the bases are always trying to relate. The codon is trying to relate with the amino acid which is going to be synthesized at the point of translation. They will always dictate the amino acid sequence in a protein. Secondly, they give information to chromosomes. They give information to chromosomes, which is then passed from parent to offspring. So the second point talks much about hereditary science, and that is the DNA itself at the point of recombination. When you talk much about the three Mendelian laws, the law of segregation, the law of dominance, they all go around about talking about the second point. So there is the law of segregation, the law of independent assortment, and the law of dominance. All those three laws of Mendel will talk about the second point. They give information to chromosomes, which is then passed from parent to offspring. At the point where it gives information to the chromosome, it means the DNA are housed by the genes. The genes are housed by the chromosome. So the genes are located in the chromosome, and in the genes, the DNA are the components of the genes. And what we talk about the genes, the genetic component, whatever is inherited, is the DNA itself. So the hereditary science is understood much by the DNA, which is a nucleic. The fourth type of biomolecules they give the chemical link between generations. So the chemical link, it still talks much about hereditary across the generations. If you want to trace one of the traits which might be seen in a family tree, we have to start from the parental generations. Parental generations, we need the genotypes. Then the first filial generations, second filial generations, to the last generation, because you have to trace. We talk about the Mendelian disorders. You have to trace or point out a gene, how it has been inherited down the line in the family tree. So it is the chemical link between generations. We are still talking much about the DNA, which is a nucleic acid. So the source of genetic information in chromosomes, I've mentioned very well. The chromosome will be housing the genes. The genes will be housing the DNA. So it's from the DNA, the genes to chromosome, chromosome to, yes. So the genes, genome, then we have, so that is the hierarchy. So they are the source of genetic information in chromosomes. Another point. So they are the most important biomolecules as well, especially at the point where we talk about genetic information, hereditary. They tell much about the protein sequence. So back to the central dogma theory. This one gives information on how important the information is passed down the line to the offspring. So it has to start at the genotypic level, the DNA itself, the central dogma talking much about that. Then it goes to transcription, then it goes to translation. At that point where you are seeing the gene product or the protein itself, then that is now when the phenotype is expressed. So it talks much about they are the ones which have to tell how the polypeptide sequence or a protein, amino acid in a protein component is going to be. So the nucleic acids, the polymers of nucleic acids are nucleotides. So you talk about a nucleotide when you have the base, you have the phosphate, and you have the pentose sugar. So the 5-carbon sugar, we talk about the pentose. And remember, in cyclic form, whatever we discuss in the monosaccharides, when you draw them in cyclic form, I said the cyclic form increase the solubility of those disaccharides. So the riboses, which form the component of the nucleic acid, have to be drawn as a furanose. They are the 5-membered ring. Remember, fructose also, the second carbon atom having a keto group, make it to be drawn as a 5-membered ring together with the 5-carbon sugars. So ribose is one of them. So there is a furanose, there has to be a phosphate, and there has to be a base. So that is a nucleotide. Nucleotides are the building blocks of the nucleic acid. So you talk about deoxyribose in the DNA case and the ribonucleic acid in the RNA. So we are going to look at the structure of deoxyribose. What do they mean at carbon atom number 2 if it's deoxyribose? What do they mean at carbon atom number 2 if it's a ribose? But then the nucleic acids are DNA and RNA. The building blocks are nucleotides. So if you talk about a nucleotide, it has a 5-carbon sugar, it has a phosphate, and it has the base. So the bases are the purines and the pyrimidines. So these are the examples of RNAs. We have the messenger RNA, which is involved much in the transcription process. There is always post-transcriptional modification, which takes place on that messenger RNA carrying the codon, where we talk about the polyadenylation at the 3' end. We have the guanosine, addition of guanosine or guanine at the 5' end, and then we have the removal of introns, the non-coding regions. Those are the 3 post-transcriptional modifications. which happen at the messenger RNA before it proceeds to synthesize protein, or before it proceeds to the translation point. So the transfer RNA is the one which carries the anticodon. The codon is carried by the messenger RNA. It has to match. So the transfer RNA carries the anticodon, the messenger RNA carries the codon. Then the ribosomal RNA is involved much in protein synthesis. So those are different types. Then the nucleotides function. The functions are many. So if you talk about the nucleic acid function, you're talking about the DNA genetic material. So sequence of nucleotides encodes different amino acids. So they always talk about the degeneracy of the codons, which you learn much in molecular biology. But for now, we know the hereditary material is only the DNA. It will be able to tell how the amino acid sequence will be in the protein at the point of translation. But remember, the central dogma theory, it has to start from the replication process to the transcription and then to the translation. The RNA involved in transcription, translation of genetic material, translation, now where the tRNA is involved, and even the ribosomal RNA fully involved in the translation process. And then the transcription, the messenger RNA, we've seen its role. But still, some viruses, they synthesize the reverse transcriptase. So like the retroviruses, the HIV virus, the genetic material for some of these viruses, the RNA itself. So those are a bit of the functions of the nucleic acid. And then we have the nucleotide. They are building blocks of the DNA and RNA. For the RNA and the DNA to be complete, it has to have a nucleotide. And a nucleotide, I still repeat, we have the 5-carbon sugar, which is the pentose, we have the phosphate, and we have the base. Secondly, they are used as intracellular source of energy, like the ATP. You've seen in the channel, I've uploaded glycolysis. Glycolysis being the simple step where glucose is broken down to those 10 steps to form the pyruvates, with the main objective of generating energy in form of ATP, or adenosine triphosphate. So this nucleotide functions as intracellular source of energy, ATP being the best example. They are involved as second messengers for most of those hormones. Mechanism of action of those hormones, which involves the second messengers, the cyclic AMP. So still the adenosine component of it there. It means they are used much as second messengers. They are also intracellular switches, where we have the G protein. G stands for the nucleotide itself. So those are just a bit of the functions of the nucleic acids and the functions of the nucleotide. So if you look at the nucleotide structure, it has to have those 3 components. We have the 5-carbon sugar, which will form the furanose ring, the 5-membered ring. It is either a ribose or deoxyribose. In the RNA, it's known as the ribose sugar. In the DNA, it is the deoxyribose sugar. But then it should also have the base, which is either purine or the pyrimidines. Those are the bases. And then it needs to have a phosphate group, that is a nucleotide. But minus the phosphate group, then we talk about nucleoside. The nucleic acid still, the RNA and the DNA. So if you see the DNA, you're talking about the deoxyribonucleic acid. It controls all the living processes, including production of new cells in the cell division. If you look at the interphase, the longest cell cycle phase, so you look at the G1, the G2, the synthesis phase, still in the interphase. In the synthesis phase, or S phase, that is where the DNA is synthesized. So production of new cells, cell division, does that? It carries the genetic code. The genetic code are the three bases, the codon we are talking much about, at the point where we have the messenger RNA carrying the codon. So the three bases represent an amino acid. Then it's going to be a length of those amino acids. We are going to talk about a polypeptide, and that will tell the sequence of a protein. So the DNA carries the genetic code. It stores and transmits the genetic information from one generation to the next. That means the hereditary science, at the point where all these phenotypes or all these characteristics are seen in the offspring from the parental generation, that is the role of the DNA. So it will transmit genetic information from one generation to the next. The chromosomes are made of the DNA. It means the DNA are housed by the genes. The genes are located on chromosomes, just as Hunt Morgan mentions that it is arranged in a linear order. The positions where the genes are located are known as the loci. So the chromosomes are made of the DNA because the DNA are housed by the genes, and the genes are located on the chromosome. So the hereditary material is the DNA. The DNA, the organelle where it is located much, 90% of the nucleus has the DNA. And remember the role of nucleus as an organelle. It is the one which controls all the activities in the cell. So that's how important biochemistry is. That's how important the organelles are. That's how important any of the metabolic pathways taking place there are. That's how important any of the processes, biochemical processes happening in those organelles are. So that's how still important to learn biochemistry. Still, when we refer to the introduction part, we'll see how important it is to learn about biochemistry so that we can understand all these processes which happens in any organism. The chemical structure of DNA versus the RNA. So we are talking about the ribonucleotides and the oxyribonucleotides. The ribonucleotides, the building blocks of the RNA. The oxyribonucleotides, the building blocks of the DNA. So look at the structure at carbon atom number 2. This is ribonucleotide because at carbon atom number 2, we have OH. That is the structure of the RNA. It is complete. This is ribonucleotide. OH at carbon atom number 2. But then for the oxyribonucleotide, there is only a hydrogen. Oxygen is missing at carbon atom number 2. So for it to be complete, we have the purine and the pyrimidine base. For the pyrimidine base, we have the pentose and we have the phosphate. So we talk about ribonucleotide. This now differentiates between the two, the ribose sugar and the deoxyribose sugar. The absence of oxygen at carbon atom number 2. The structure is simple, but then at carbon atom number 2 of ribose or RNA, we have the ribose, presence of OH, but then deoxyribose, we have the absence of oxygen. So that is how the name goes. They are two related pentose sugars. RNA contains ribose, DNA contains deoxyribose. Then we were talking about the bases, the purines and the pyrimidines. For the purines, you can see the double ring around them. So the purines, we have the adenines and the guanines, and you can differentiate between the two. The difference lies on the adenine. It has an amine group up there, while for the guanine, it has a keto group up there. And then for the guanine, there is also an amine group down here, while it is missing in adenine. But remember, around the carbons, balance your bonds around the carbons to be 4, around the nitrogens to be 3. If you master the atomic configuration, electronic configuration, it can be very, very easy. Electronic configuration, 2 to 5 for nitrogen, 2 to 4 for carbon. The carbon will need 4 electrons on the outer shell to be stable. You'll be good to go. Nitrogen needs 3 electrons on the outer shell for stability. You'll be good to go. So these are the examples of purines, adenine and guanines and the purines. Then the pyrimidines, we have uracil in RNA, thiamine in DNA, but then the 3 pyrimidines are cytosine, thiamine, and uracil.